Minnesota's sulfate standard has a long history. Dr. John Moyle from the Minnesota DNR performed a series of field surveys across Minnesota in the 1930s and 1940s. Based on this research, Dr. Moyle stated that wild rice was generally absent when sulfate levels were greater than 10 milligrams per liter. While scientists did not agree on a correct number, all agreed that if sulfate was indeed affecting wild rice, it was during early life stages. Prime wild rice habitat in the state is primarily in our Laurentian mixed forest biome, and also some scattered populations within the eastern broadleaf forests. There is little to no rice in the prairie pothole region of southwestern and far western Minnesota. These areas to the west and southwest have higher naturally occurring levels of sulfate. In order to better understand the effects of sulfate on wild rice seedlings, hydroponics, or water-only testing, was conducted at the University of Minnesota Duluth on behalf of the Minnesota Pollution Control Agency. This testing, as well as testing conducted by Fort Environmental Laboratories, showed that sulfate was not toxic to wild rice until levels reached 1,600 milligrams per liter, about 160 times the current standard. However, another theory was put forth, contending that sulfate itself, while not toxic, could be converted to sulfide in the sediment, which is toxic. Sulfide occurs where there is little or no oxygen present. Additional research was conducted at the University of Minnesota Duluth to measure the effect of sulfide on wild rice seedlings. This research took place in containers with no oxygen and enhanced sulfide concentrations. The results of this testing showed that wild rice seedlings were affected by sulfide at fairly low levels. However, only the green parts of the plant were affected. The roots and the seeds, which exist in the sediment where no oxygen exists and where sulfide would be in a natural environment, were not affected. Sulfide does not normally occur when oxygen is present, and the green parts of wild rice are not normally exposed to sulfide. In response, additional testing was conducted at Fort Environmental Laboratories. This test design used nitrogen gas to remove oxygen from the water and keep the sulfide concentrations constant. This allowed only the rooting portions of the plant, the roots and the seeds, to be exposed to sulfide, and the rest of the plant, the green parts, would be exposed to oxygen, as they would be in nature. This research revealed two very important findings. First, sulfide is much less toxic to wild rice than was previously thought. And second, addition of iron mitigated sulfide toxicity even more. In fact, the results of the research exposing only those portions of the plant that would normally be exposed to sulfide were approximately 10 times higher than the research previously conducted.